Okay, so welcome to this third video on Edman degradation. So far what we've done is, just to recap, is we've taken this polypeptide here and we've reacted it with phenyl isothiocyanate, and that forms this molecule here. Then what we did is we put it in the presence of a strong acid like hydrochloric acid, and that triggered this reaction of this molecule into this molecule. Then this molecule collapses into this molecule here and the polypeptide minus the first amino acid. So what we're now going to do is we're going to use this molecule here in order to analyze what that first amino acid was, because as you can see, this molecule is going to depend on what amino acid was in that first position, because we have this R group here. So it's going to be different depending on what the amino acid, what that first amino acid in the polypeptide was. And therefore we can use the different chemical properties of this molecule to determine what that first amino acid was. Okay, so you don't quite use this molecule to analyze what the first amino acid was. Instead, you firstly change it into another molecule. So I'm going to redraw that one out, and then we'll see what we changed it into. So let's just copy this molecule out. You have your phenyl group here, and then you have this nitrogen bonded to the hydrogen. Then you have your carbon, and then this 5-membered ring, nitrogen here, the sulfur down there, carbon at the bottom there, carbon here, and then off this carbon you have a hydrogen and the R group, and then a carbon R group there. Right, so we're going to turn this molecule into another molecule which we can then do chemical analysis on to try and work out what this R group is, basically. Okay, so the reaction you're going to do is you're going to basically you're going to break this 5-membered ring apart and make a new 5-membered ring. So you're going to break this bond here, and you're going to make a new bond between this carbon here and this nitrogen here to make a new 5-membered ring, basically. Then what you're going to do is clearly you're going to have to take this hydrogen off here, so that bond's going, and you're also going to have to do something about this sulfur. So what you do is you forge a double bond here to replace the bond that the sulfur has lost, basically. But then this carbon's going to have 5 bonds, so you break one of the bonds there and put the hydrogen on that nitrogen. Okay, so let me try and show you this. Alright, so let's put that phenyl group back where it was. So here's the phenyl group. We'll leave that nitrogen. We'll try and leave everything as much as possible in the same position. So let's put that carbon in the same position. Then it's now going to be double bonded to this sulfur. So you've broken this bond between this sulfur and this carbon, and now this sulfur is going to double bond with this carbon. That's going to result in this nitrogen now only being linked by a single bond. And the drawing is going to look quite odd because it's going to be a very odd. It's going to be a very odd notation for a 5-membered ring. Usually, it's customary to write 5-membered rings in this nice pentagon shape, but we're not going to end up with that in this picture. Okay, now this carbon we haven't done anything with, so let's just replace that back in. So let's put this carbon with its hydrogen and its R. All I've done is flipped it upwards. Right, this nitrogen now takes the hydrogen from this nitrogen that had to lose it because it's forged this new bond with this carbon, and that is basically going to be there, basically. Okay, now we have this carbon here, so let's put this carbon here, and this carbon now has a bond to this nitrogen here, and a carbon R group like that. So basically you have reforged a 5-membered ring here, but it's different from the original 5-membered ring you had. And it is this molecule here that you now use to analyze, basically. So you analyze this molecule, okay, and you can basically use chemical reactions to determine what this R group is. So you can determine what the R group is there, and that tells you what the first amino acid in your protein was, basically. However, we know that, in this reaction, at this stage, it gave you off the rest of the polypeptide chain. So all this basically did was it used the first amino acid here to make this molecule here. But the rest of the polypeptide is completely unharmed, basically. It's here. So now what we can do is we can perform the whole process over again. That will tell it. Then what we can do is we can work out what the second amino acid is. And then when we do the whole process again, what you'll end up with is another the fragment of the polypeptide after that cleaved off. So you can then do Edman degradation again to work out what the third amino acid is, and you can continue this process on, doing this reaction over and over again, and find out what each of the amino acids is in the polypeptide, and that way you can sequence your polypeptide. You can find the sequence of amino acids that comprises it. Okay, so that is Edman degradation, and as you can see, it is a method for analysing what the amino acid sequence of a polypeptide is.